Now I'd like to start to describe the particulars of the senses. Now, if you stop pretty much anybody on the street and you ask them what are people's senses, they're going to usually respond with what we consider the big five senses: touch, taste, smell, sight, sound, or vision and hearing. That's not a real accurate description of the senses that we have. Various sources are going to give you different lists of the types of senses that are available. At one time I heard that there are 17 different senses. Part of that comes from these five senses that we're talking about right here really include multiple senses. We're going to start by talking about taste, and we'll see that there are actually five separate tastes. They don't interact together other than the fact that they're all localized to the tongue. Likewise, there are different aspects of touch, different aspects of vision, etc., etc. So there are a bunch of different types of senses. I'll sometimes use the term sensory modality. That's just a fancy way of saying sense. To some degree, it's just something I'm comfortable with saying, having talked about senses for a long time through my schooling and my career. If I say sensory modality or sensory submodality, I'm really just referring to one of these senses. A submodality would be a particular type of one type of sense. So there are five submodalities of taste, for instance. Don't get caught up in these words. They're just words that I'm comfortable using quite a bit. So just when you hear modality or submodality, just plug in type of sense when you're thinking about that.
Sensory systems are associated with receptive surfaces. For example, taste is associated with the tongue. If you look at the tongue with the naked eye, you can see a number of bumps all over it. Those bumps are referred to as papillae, plural for the word papilla, which means little bump. Within each papilla are taste buds, which are indicated in the picture in the lower right corner, and were described earlier in terms of the specialized receptor cells. In describing transduction of the taste submodalities, there are five, as you see listed here. Probably a few decades ago, the list would have only been four. The fifth one has only relatively recently been accepted as a taste modality. It was originally discovered in the early 1900s, the first decade of the 1900s, but it took a good 75 or 85 years before it was accepted into the level of understanding that would make it into textbooks. It's possible in the future that this list could get longer, as there is some new evidence coming to the forefront of a possibility of a new taste. Personally, I don't believe that's going to be the case, and that's actually a topic I'll discuss within the digestive system. Possibly the most straightforward example of transduction of any sensory modality is the sense of the salty taste. Table salt is sodium chloride, but really the saltiness that we taste from sodium chloride is strictly because of the sodium ions. And the salty taste could be described simply as our perception of the sodium concentration in our saliva. As I've indicated here on the side, the sodium ion with brackets around it means concentration of sodium ions. And if you think back to how electrical activity works across the cell membrane, as in terms of membrane potential, the first step in changing membrane potential is usually the movement of sodium ions across the cell membrane. And really all that's happening in the salty taste is that as you eat something with more sodium ions in it, most likely in the form of sodium chloride, the concentration of sodium ions will increase in the extracellular space, i.e. where your saliva is, and the driving force of sodium ions entering into the cell will increase. The greater that concentration gradient, the greater the movement of sodium ions into a cell. And so the taste receptor cells are depolarized by the entry of sodium ions into the cell. If you consider the stimuli that we consider sour, you'll notice that they're all really just acidic compounds. Citric acid, acetic acid, as in vinegar, those sorts of things taste sour. And really sour is our perception of the hydrogen concentration in our saliva, much like saltiness is sodium concentration. Now, the idea of hydrogen ion concentration in a solution has a very specific meaning. That is the pH. So as our saliva becomes more acidic, as the pH goes down because hydrogen ion concentration goes up, that means that the hydrogen ion concentration gradient will increase and will have the same effect as the sodium ion concentration has for the taste of saltiness. As far as membrane potential is concerned, sodium ions and hydrogen ions are not much different. They're both a positive ion. So if a positive ion crosses through the cell membrane, it's going to depolarize the cell, leading to a membrane potential change. So really sour and salty aren't essentially any different. It's just a matter of which ion channels are present in those particular gustatory receptors. Now, the other taste stimuli I want to describe are a different type of stimulus. Salty and sour were both really just sensing the concentration of particular ions in the saliva. The rest are all based on the idea of a particular molecule or type of molecule binding to a receptor on the surface of a gustatory cell. This is really how all of the rest of chemical sensation is based. Not just these three taste stimuli, but also olfactory, or the sense of smell, nociception, or the sense of pain, and to a large degree, all of the chemical interactions that happen in our body, whether it's neurotransmitters, hormones, or cell-to-cell interactions, besides the two that I've already described, everything else is really just about molecules interacting with receptor proteins within the target cell. And for the sense of sweet, the receptor protein is essentially the glucose receptor. If you zoom in on the three figures next to the word sweet, you'll notice that those are just chemical representations of the glucose, fructose, and galactose molecules, which are the three hexoses, the three simple sugars, monosaccharides, that we find in most carbohydrate-based nutrient sources. They all have very similar structure. There's slight differences, but they're all based on the same chemical formula, which is C6H12O6. And they're all arranged in a ring-like structure. The rings are slightly different, but they're similar enough. And they all interact with the same protein receptor. And so their presence in our saliva, because they've dissolved into it from whatever food we're eating, will interact with the sweet receptor. It is possible, I'm not familiar with it myself, but the three different monosaccharides that we're considering here probably have different affinities for the receptor, so they might have different binding capabilities and therefore taste sweeter or less sweet relative to each other. And these three molecules are not the only ones that bind to the glucose receptor, or the sweet receptor. Anything that can bind to that receptor would be considered sweet. And the whole idea behind artificial sweeteners or low-calorie sweeteners is that there are molecules that bind that just don't have a caloric content the way that these carbohydrates do. So Splenda or Truvia, saccharin, all of those molecules are simply artificial sweeteners because they have the proper shape to interact with the sweet receptor on our tongue. However, once they get into our digestive system, they do not get digested chemically the same way, and they have no real caloric content to them. The taste of bitter is really the taste for molecules that are collectively known as plant alkaloids. And it's not usually a pleasant taste, and it's mostly associated with stimulating a gag reflex. The reason for that is that these types of molecules are found commonly in plants and are associated often with other types of molecules because they also come from plants, which are potentially toxic to our body. Now, the picture I have here is just an image I got off the internet of a vegetable called a bitter gourd, and it's probably just referred to that because it has a high concentration of these bitter tasting molecules, plant alkaloids, in it. Now, there are a number of things that we consider bitter in taste. Oftentimes we will eat those foods despite the bitterness because, one, we know collectively, culturally, that they're not toxic foods. They just happen to have this type of molecule in them. And we often mask that bitter taste with something stronger, whether it's putting cheese or broccoli, cheese on broccoli so that it tastes, or it has more of a fatty content to it, or putting milk or sugar in coffee so that it tastes sweeter, whatever it might be. We can cover the bitterness and make those sorts of foods palatable, even though they have a stimulus in them that's often going to fundamentally activate a gag reflex because of our inherent need to keep potentially toxic plant things out of our systems. Generally speaking, tastes are not specifically mapped to a particular part of the tongue. Even though you might hear that from some sources, it's not really a truth that you could say. It's a misconception that's been propagated by certain sources. Bitter receptors, however, are likely to be more commonly found towards the back of the tongue, or more densely populated. They'll be found all over the tongue, but the most bitter receptors are going to be in the back, sort of as a last chance to stimulate a gag reflex before we swallow something. The fifth taste, which I mentioned earlier, was originally discovered early in the 1900s. I believe 1906 is the date of the paper published describing it, is formally called umami, which roughly we can translate to mean savory. It's essentially the taste for amino acids, and much like monosaccharides bind to the sweet receptor or plant alkaloids bind to the bitter receptors, amino acids bind to the umami receptors. The picture I have here, again, I just got it off the internet, is a picture of a number of bones that have been prepared so that the very proteinaceous bone marrow is the food that's described here. I could have just as easily put a steak, I suppose, but for some reason this image just seemed to say umami to me. Now, the word umami is a Japanese word. The original scientists that discovered this taste were Japanese, and as is common in science, whoever discovers something gets to name it. It's in fact a word that they invented for this, which essentially translates to tastes good, which is one meaning of the word savory, I suppose. But really, when we describe savory in a culinary sense, we're often describing proteinaceous or meat-based foods, and that's largely where umami taste stimuli are going to come from, because meat is often the most proteinaceous food that we ingest. The next of the major senses that I want to talk about is the olfactory sense, or the sense of smell. I'm not going to describe it in the same kind of detail as taste, to take apart the different submodalities within it. Partly that's because taste is much better understood than smell. There might be various reasons for that. My best understanding is really that the sense of smell is a very hard stimulus class to control, to be able to elicit responses that can be studied in a laboratory setting the same way that taste stimuli could be studied. Nowadays, technology probably makes it a little bit easier to expose a subject to odor molecules, odorants, as it were, but then a taste molecule. You can easily put drops of a sugar solution on the tongue and test the reactions to taste buds to that. And for the olfactory sense, it's just historically harder to do that. Nowadays it might be easier, and nowadays there may be better data coming out describing the sense of smell. However, it's not as well described as the sense of taste. Another possible reason for that is because the sense of smell is very, very closely integrated directly with the cerebrum. The other senses are going to go through some sort of brainstem connection usually. Vision's a little bit different than that too, which I'll describe shortly, but the olfactory epithelium, the external receptor surface where the receptor cells are found, project directly through the cribriform plate of the ethmoid bone into the olfactory bulbs, which are on the ventral surface of the prefrontal cortex, and they then connect back to the posterior prefrontal cortex and some areas of the limbic system and medial temporal lobes, having a very direct connection to very high centers of the brain, which are often hard to study in a neuroscience lab just because they're very complicated. So there's just not a whole lot to say about olfaction as specifically as the 5 submodalities of taste that I just described. I do want to mention, however, one way to consider the sense of smell is to consider which direction the odor molecules are passing over the olfactory epithelium. When we inhale through our nostrils and the air moves back posteriorly through the nasal cavity, that's described as an orthonasal presentation, which means that the odor molecules move from anterior to posterior across the epithelium. And that's how we often think primarily smells. However, the sense of smell, olfaction, is also closely tied into our experiences of food. If you think about the tastes that I just described, really not all of them are pleasurable experiences. And depending on the type of food you're talking about, most of them wouldn't be. You probably wouldn't want savory tastes tied up in a candy bar necessarily. Or sour is not always a pleasant experience. You wouldn't want sweet to be part of a savory meal all the time. But you only have those 5 submodalities of taste to work with, which would make food very boring if it was just those 5. And so most of our enjoyment of food is really from our olfactory sense. And when that happens, it's partly from an orthonasal presentation of food as it's coming closer to our mouth, but also as we're ingesting the food and breaking it down in our oral cavity, some odor molecules will be released from the food, and sometimes only released from the food when it's being chewed up, which will then pass through the pharynx up into the posterior nasal cavity and reach the olfactory epithelium in a reverse direction, or retronasal presentation. There's an article I read a while back, and I can't recall the citation exactly anymore, but it described this idea in terms of coffee. Smelling coffee while it's brewing or while it's in your cup can be a very pleasurable experience because of the odors associated with coffee. But then drinking coffee for many people is an unpleasant experience because coffee itself has such a bitter component to it, most of which is in the taste, but then also the retronasal presentation of those same odors is often perceived differently. And a lot of that can be mitigated by adding lots of sugar or cream or something like that to the coffee so that those basic tastes overwhelm the retronasal presentation of the coffee. It's not as fundamental to smell necessarily as the 5 submodalities of taste are to that type of sense. But it is important to consider anatomically that smells can be presented to the olfactory epithelium from 2 directions, either orthonasally through the nostrils or retronasally from the posterior direction, what's called the nasopharynx. If you consider the transduction mechanisms involved in olfactory senses, they're all essentially the same. Odor molecules, odorants, are airborne, and we inhale them through our nasal cavity. They dissolve into the nasal epithelium, and they interact with protein receptors, olfactory receptors, on the olfactory receptor neurons. The olfactory receptor neurons are neurons. They are essentially free nerve ending type receptors. The dendrites have tendril-like extensions out into the nasal mucosa or mucus layer, and the receptors are in the cell membrane there. The cell is a bipolar shape, and the action potential that's generated by the stimulus interacting with the receptor protein generates an action potential that travels from the nasal epithelium across the cell body and through the axon, which is going along the first cranial nerve, the olfactory nerve, and synapses in the olfactory bulb on the other side of the cribriform plate. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer.